representing the sample input. However, depending on the type of NGS application, different library preparation steps are taken. There are 4 types of NGS applications which are considered below: whole genome sequencing, exome sequencing, library preparation kit, ideal for any genome where there is 1 to 2 micrograms of genomic DNA available. The purpose of this kit is to avoid PCR amplification errors over long distances. Genomic DNA is isolated from the sample and fragmented physically or chemically, often creating overhangs. The resultant DNA fragments are then purified using magnetic beads with a desired size of 350 or 550 base pairs. This occurs by incubating specific platforms available on the market are very accurate, they are still prone to error. Even at accuracies of 99% and greater, a sequence generated may contain incorrect nucleotides. This means that if a machine's accuracy is 99%, 1 base pair is read incorrectly out of 100. Since NGS platforms generate high amounts of output, these errors can add up quickly. The way to circumvent NGS platform limitations is to sequence nucleotides multiple times. The number of times a nucleotide is sequenced is referred to as coverage or depth. Coverage per genome can be calculated by dividing the total output generated specifications. Having either more or less DNA results in less efficient sequencing reaction runs. This generates low-quality data due to read problems from slow cell saturation or reduced coverage because of insufficient DNA. In terms of quality, a good quality library is one that has a diverse set of DNA fragments with minimal duplicate fragments. This is important because during PCR amplification, duplicates of fragments will be generated. The consequence of duplicate fragments is that the sequencing reaction will be biased towards these fragments. Rather than having a wide range of fragments sequenced, the same fragments are sequenced repetitively, resulting in overrepresentation in the machine output. Library quantification fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slide gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past. It isn't capable of generating nearly as much output as the Illumina high-seq machines. The ion proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybrid
based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read,
Myself Sarita, I have done PhD in biotechnology, and here I came for the NGS workshop. Here we got knowledge about here we got a good knowledge about the next generation sequencing, which included DNA sequencing, RNA sequencing, and somewhat this workshop also covered this CRISPR-Cas9. So this was overall a good experience for us to learn. So we will.
Yeah, myself Sonika Agrawal, and I am pursuing my PhD from National Dairy Research Institute (NDRI). For me, it was very good because I am very new for this technology, and I am working for this next generation sequencing for my project. But I don't know nothing. But when I am going from this workshop, I learned so many things. So I am very happy to join this such type of workshop. So thank you.
Hello. My name is Sonia Arora. So I've done PhD in microbiology, and I've come from Ghaziabad for this workshop. And overall experience for this workshop was very good. Like, I learned various technologies like RNA sequencing, DNA sequencing. Get some overview about this CRISPR-Cas9 new technology, and so it was an overall new experience for me to learn this technology. And although it was a very short, limited period by workshop, but it was very good, and I hope I'll be continuing this relationship with the BDG LifeSciences in future also. Thank you.